In the early 1900s, an immunologist named Paul Ehrlich popularized the fantasy that one day scientists will be able to construct drugs that only target the specific disease-causing organisms or molecules. This would mean that treatments such as chemotherapy will no longer attack and kill both the cancer cells and our normal healthy cells. This fantasy was called the magic bullet, and it would be an invaluable breakthrough in medicine. A century later, monoclonal antibodies are now showing that this dream was not just a fantasy, but it is slowly becoming a reality. So what are monoclonal antibodies? Well, as the name suggests, monoclonal antibodies are, in fact, antibodies. Antibodies are these Y-shaped proteins inside our bodies that serve as part of the immune system. Each antibody will have a slightly different shape on its end that recognizes and binds to specific disease-causing molecules called antigens. There are 4 different types of monoclonal antibodies that are being used in medicine. The first ones made were called murine-based monoclonal antibodies, and these antibodies were obtained by extracting the immune cells from an immunized mouse and collecting the antibodies they produced. However, when these were given to patients, it often led to severe allergic reactions and rejection because they came from mice. This led to the development of chimeric and humanized monoclonal antibodies that were a combination of a mouse part, as seen in yellow, and a human part, as seen in red. Since they were mostly human in origin, this significantly reduced the side effects experienced. The greatest achievement came with the formation of fully human monoclonal antibodies. When these were injected into the patients, they produced the least side effects. So what are monoclonal antibodies actually used for? Well, in the last decade, the range of their uses has exploded, and they're currently used in fields ranging from breast cancer, melanoma, lymphomas, autoimmune diseases such as rheumatoid arthritis, Crohn's disease, asthma, and MS, several cardiovascular diseases, viral infections, diagnostics, and are being developed for many more applications. Every monoclonal antibody is different and binds to different antigens. As a result, they generally work in different ways for different diseases. However, I'll explain 2 general ways that they have been shown to work in treating cancer. The immune cells in our body usually do a pretty good job at detecting foreign invaders. However, sometimes they are not that good at detecting our own cells that turn bad, such as tumors. A monoclonal antibody called rituximab can recognize and bind to these tumors. When the drug binds to the tumor, it can alert the rest of the immune system. The immune cells know where the cancer is, they go and bind to it, and cause a series of events that leads to the tumor cell death. Our bodies naturally produce chemical signals that can bind to receptors and tell our cells to grow and divide. However, some cancer cells, such as breast cancer, produce too many of these receptors and they divide uncontrollably. A monoclonal antibody called trastuzumab can bind to these receptors and stop the growth signals from binding. This stops the uncontrollable cell division and is used to treat breast cancer. The future for monoclonal antibodies looks good, with over 300 different antibodies currently in development. One problem that they face at the moment is that they cannot cross the blood-brain barrier, and therefore have little ability to treat neurological diseases such as Alzheimer's. However, research into different ways of allowing the antibodies to cross the barrier, such as using liposomes, is being developed and showing promising signs. Another method of using monoclonal antibodies to treat cancer that is sparking interest involves attaching the antibody, such as abritumab, to a toxic substance. This complex will then travel around the body and bind to the specific antigen that the antibody recognizes. The toxic substance may be a radioactive isotope that will directly target the tumor cell due to its close proximity. Or the toxin may be another type of chemotherapeutic agent that detaches from the antibody and directly attacks and kills the cell. These are just some ways that monoclonal antibodies are used. The future looks exciting, and we are getting closer and closer every year to making these magic bullets.